Hello everyone, in this video I am going to explain about synapse. The things which I am going to cover in this presentation about synapse are the definition of a synapse, the different types of synapses, and the process of synaptic transmission. So here are the definitions of synapse. We can define synapse in number of ways. So the basic definition of a synapse is nothing but it is a junction between two neurons which are nothing but nerve cells. So in a simple way we can define synapse as the junction between two nerve cells or neurons. This word synapse was introduced by Sherrington. Synapse is also otherwise called as neuronal junction. It is actually the site of transmission of nerve impulses, and this site can be a site between two neurons or the nerve cells, or it can be the junction between a motor neuron and a muscle cell or an effector cell, or else it can be a junction between a neuron and a gland cell. In another way, we can define synapse as a junction between the presynaptic neuron and the postsynaptic neuron. So what are presynaptic neurons and postsynaptic neurons? As already said, synapse is nothing but a junction between two neurons, and the neuron before a synapse is nothing but a presynaptic neuron, and the one which is present behind the synapse is nothing but the postsynaptic neuron. So synapse in another way we can define synapse as the junction between the axon terminals of one neuron, that is the presynaptic neuron, and between the dendrites of the second neuron, that is the postsynaptic neuron. Anatomically, if we see, this junction between two neurons has been categorized as follows. The junction can be of axodendritic type or axosomatic type or axonomic type, depending upon the junction formed between the different parts of the two neurons, that is the presynaptic neuron and the postsynaptic neuron, based on the part of these two neurons which are taking part in the formation of a junction, which ultimately constitutes a synapse. If at all the junction is between an axon of one neuron and the dendrite of another neuron, then that type of synapse is axodendritic type. If the junction is between the axon of one neuron and the cell body of another neuron, that is nothing but the postsynaptic neuron, then that type of synapse is nothing but axosomatic type. If at all the junction is between the axon of one neuron and the axon of another neuron, which take part in the formation of junction, then that type of synapse is nothing but axonomic type. So these three are the different types of synapses based on the parts which take part in the formation of synapses. Anatomically speaking, synapses are categorized as exodendritic, axosomatic, and axonomic based on the parts between which the junction is formed. So before going further, we have to know about synaptic cleft. So what is cleft? Cleft is nothing but a space or a gap. So as the name indicates, synaptic cleft is nothing but the gap between two neurons at a nerve synapse. Synapse is the junction between two neurons. The one being the presynaptic neuron, the first one, and the another one being a postsynaptic neuron. So the gap which exists between these two neurons which form a nerve synapse is nothing but synaptic cleft. It is nothing but the gap that is present between neurons at a nerve synapse. So next, coming to types of synapses. Synapses, physiologically if we see, synapses are of two types. First is chemical synapse type, second type is the electrical synapse. Coming to the first type, that is chemical synapse. In occurrence, if we see, this type of synapses are more in number. As the name indicates, this type is named as chemical synapse. So here the transformation or of the information is through chemicals. That is why this type of synapse is called as chemical synapse. Here this type of synapse, they transfers the information through the help of chemicals. So what are actually these chemicals? The chemicals which take part in the transformation of nerve impulses are nothing but neurotransmitters. Here are some of the well-known neurotransmitters: acetylcholine, serotonin, dopamine, etc. So to remember, here are some of the well-known neurotransmitters which take part in the chemical synaptic transmission. These neurotransmitters, they are released from specialized structures which are nothing but synaptic vesicles of presynaptic neurons, and as soon as they are released, they binds to the specific chemoreceptors of postsynaptic neurons, thus help in the transmission of nerve impulse. So as a result, what happens? When these neurotransmitters, they bind to the specific chemoreceptors of postsynaptic neurons, they results in the opening of the sodium ion channels and they generate a nerve impulse. If we see the mode of transmission, it is very slow and steady in case of chemical transmission through a synapse. Chemical synapses, they transmit the signal only in one direction. That is, they are unidirectional in their nature. So if we talk about a chemical synapse, here the transmission of the information or the signals is only in one direction. Coming to the second type of synapse, electrical synapse. So physiologically there exists two types of synapses. One is electrical chemical, and the second one is the electrical synapse. As the name indicates, here the transmission of the information is through electrical signals, not through chemicals. And these synapses, they are fewer in number and they transmit electrical signals. They transmit the information in the form of electrical signals. In this type of synapse, two neurons are connected by channel proteins which allow the direct passage of ions and signaling molecules. There is no chemical in between the two neurons. So the transmission is faster in case of chemical synapse, as there are no receptors taking part in the transmission of a nerve impulse. If we see the nature, these electrical synapses, they transmit signal in both the directions. They are bidirectional in nature. Previously, the chemical synapse, we have seen that they transmit signal in only one direction, whereas electrical synapses, they transmit signals in both directions and they are faster. The transmission is faster in case of electrical synapse, as there are no receptors. This is the important one. Because of the absence of receptors, the transmission in case of electrical synapse is faster. So here in this figure you can see the two different types of synapses. First one is the chemical synapse and the second one is the electrical synapse. Here this structure is nothing but the presynaptic neuron, and the below one is the postsynaptic neuron. The same is the case here also. This one is presynaptic neuron, this one is postsynaptic neuron, and the membranes at the edge of these neurons is nothing but the membrane which is at the terminal part of this presynaptic neuron is presynaptic membrane, and here this is the postsynaptic membrane of postsynaptic neuron. In this figure, here these vesicles are the synaptic vesicles which release the neurotransmitters. As I said, in chemical synapse, these neurotransmitters, they bind with special receptors present on the postsynaptic membrane of postsynaptic neuron. So here the transmission is unidirectional. Only in one direction the transmission of signals takes place, which is represented here by means of this arrow, unidirectional. So as soon as an action potential reaches this presynaptic neuron, the synaptic vesicles, they release the neurotransmitters. These neurotransmitters they bind with the receptors present on the postsynaptic neuron, and this results in the transmission of information. If we see the electrical synapse, here we don't see any chemical. So as soon as an action potential reaches the presynaptic neuron, the terminal, this part is the terminal knob of presynaptic neuron. Here these specialized structures are nothing but the gap junction channels. So through these gap junction channels, directly the transmission of information takes place. And as there are no receptors, here you can see the presence of receptors in chemical synapse. whereas the chemical electrical synapses do not have any receptors. So the transmission here is, it is slow and steady, whereas it is faster in case of electrical synapse, as they don't have any receptors which we can see in chemical synapse. So through these gap junction channels directly the information is, there are no receptors. So the movement of ions is through these gap junction channels, and as such the information is transmitted directly without the involvement of any neurotransmitter or any receptor. So coming to the synaptic transmission, how actually the transmission takes place through a synapse. And here I will discuss about the chemical transmission, not an electrical transmission. That is the transmission through a chemical synapse, not through an electrical synapse. So if we see the process of synaptic transmission through a chemical synapse, what happens actually in this process of conduction of information? The action potential, as soon as it reaches or arrives at the terminal knobs of presynaptic axons, it depolarizes the presynaptic neuron membrane. As a result, the voltage gated sodium channels present on the presynaptic membranes, they open. This results in further depolarization of the presynaptic membrane, and this causes voltage gated calcium channels to open. When the calcium voltage gated calcium channels open, the calcium ions flow into the presynaptic cell or neuron because its concentration is greater outside the cell, that is the presynaptic cell, than inside it. As a result, the synaptic vesicles which are present in the as a result, the synaptic vesicles which are present in the presynaptic neurons, they migrate towards the presynaptic membrane and they merges with it and they forms a continuous membrane, releasing the neurotransmitters. So neurotransmitter is released by the synaptic vesicles. Where it is released? It is released into the gap which is present at the site of nerve synapse. So they release the neurotransmitters into synaptic cleft, and by a process which is nothing but exocytosis. So by the process of exocytosis, the synaptic vesicles present in the presynaptic neurons, they release neurotransmitters into the synaptic cleft. When these neurotransmitters are released into the synaptic cleft, they diffuses across the synaptic cleft. They binds with the receptor proteins which are, or the chemoreceptors present on the postsynaptic membrane. As a result, again the sodium channels they open and depolarization takes place, and this starts the transmission. So here is the process of synaptic transmission with the help of a figure. So if we see this figure, here the structure of a chemical synapse is clearly shown, as well as the process of synaptic transmission is also shown. So this is presynaptic neuron, and this terminal part of this presynaptic neuron is the terminal knob, and from here this part is nothing but the membrane of postsynaptic neuron. So the junction present between presynaptic and postsynaptic neuron itself is synapse, and the gap, this gap which is present between presynaptic and postsynaptic neurons, or the gap which is present at a nerve synapse, is nothing but synaptic cleft. So if we see this terminal knob of a presynaptic neuron here, so this is the presynaptic terminal knob. In this region we can see these vesicles. So the first step in the synaptic transmission is neurotransmitter is synthesized in cell body or in the terminal knob. So here it is neurotransmitter. Through biosynthetic enzymes it is synthesized and stored here. So second step is neurotransmitter is packed into the vesicles. It is synthesized here and packed in these vesicles. So these vesicles are filled with neurotransmitter molecules. So this is a cyclic process. Firstly, through biosynthetic enzymes, these neurotransmitters are synthesized and they are packed into these vesicles. So here we can see the molecules, neurotransmitter molecules. Third step is neurotransmitter is released when vesicles fuse. So when the action potential is released and when the voltage gated channels are opened and further depolarization takes place, and as a result these vesicles which have neurotransmitters in them, they fuse with the membrane, this membrane of presynaptic neurons. So when they fuse with this, they merge with this membrane and they finally release these molecules of neurotransmitter into the synaptic cleft. So here the next step is this synaptic vesicles, they are merging with the presynaptic membrane and they are releasing. Here in this figure it is shown with the help of arrows the release of the neurotransmitter molecules into the synaptic cleft. So the next step is fourth step. Till here this was the first, this was second, and this was the third. So here as soon as this neurotransmitter is released into the synaptic cleft, what happens? It binds to and activates the postsynaptic receptor. So 1, 2, 3, these are nothing but the receptors. So these molecules, they bind to the post receptors of the postsynaptic membrane and activate the receptors. So here next, neurotransmitter, it diffuses away, it won't stay too long here. These neurotransmitters, they won't stay too long in this cleft, but what happens is these neurotransmitter, it diffuses away and is metabolized and or it is transported back into the terminal. So here as soon as they bind to the receptors of postsynaptic membrane and they activate them, then soon they diffuse here in the cleft, synaptic cleft, and again they are transported back through these transporters into this terminal knob of presynaptic neuron. So here again so this is a cyclic process. It started with the biosynthesis of neurotransmitters, the packing, then merging or the fusion, here we can see, and they become a continuous membrane, release the neurotransmitters, and these neurotransmitters they bind with the receptors of postsynaptic membranes and they activate them, and soon this neurotransmitter it diffuses away and is metabolized or is transported back into this terminal knob. So again the process is continued. So when these receptors they bind to the postsynaptic membrane, they initiate a nerve impulse by depolarizing this postsynaptic membrane. So this is the process of synaptic transmission from one neuron, that is presynaptic neuron, to a postsynaptic neuron, and this transmission is in one direction only. The transmission of information takes place from presynaptic to postsynaptic neuron only. It is in one direction only. It takes place only in one direction. So a synaptic transmission through a chemical synapse is unidirectional, that is through it passes from presynaptic to postsynaptic neuron only. So this is about the synaptic transmission. Before concluding, I'll give a quick recap of what I have explained till now. So synapse is nothing but a junction between two neurons. In simple way, we can define synapse as junction between two neurons, otherwise called as neuronal junction. In another way, we can define synapse as the site of transmission of nerve impulses between two neurons, or between a motor neuron and a muscle or an effector cell, or between a neuron and a gland cell. Anatomically, if we see, the junction between two neurons can be between an axon of one neuron, that is presynaptic neuron, and the dendrites of the second neuron taking part in the formation of a synapse, that is nothing but postsynaptic neuron. So this type is axodendritic type. In axosomatic type, the junction is between axon of one neuron and cell body of another neuron, whereas in axonomic type, the junction is formed between the two axons. So this axon is of presynaptic neuron and this axon is of postsynaptic neuron. So this is the anatomical classification of synapses. Anatomically, if we see, they are of the following three types, that is axodendritic, axosomatic and axonomic. Now synaptic cleft, the gap which is present between the two neurons at a nerve synapse, is nothing but the synaptic cleft. Types. Physiologically, if we see, synapses are of two types, chemical and electrical, and this I would like to explain with the help of this figure. It is clearly shown here that this is a chemical synapse which has neurotransmitters in it, whereas in electrical synapse there is no chemical involved in it and there are no receptors also. Here the transmission is unidirectional, here it takes place in both the direction. Here the transmission is slow and steady, here it is faster. This type of synapses, they are about 80% of the total synapses, they are more in number, whereas these electrical synapses, they constitute only 20% of the total synapses and they are fewer in number. As these electrical synapses, they do not have any receptors which we can see in chemical synapses, the transmission here is faster when compared to the chemical synapses. So these two are the presynaptic neurons and here these two are the postsynaptic neurons. And here in electrical synapse you can see the presence of gap junction channels. So coming to the last thing, that is the synaptic process of synaptic transmission. As I already mentioned, here this is a synaptic transmission through a chemical synapse. As soon as the action potential reaches the terminal knob of the presynaptic membrane, here we can see the synthesis of neurotransmitters, packing of neurotransmitters into the vesicles, and these vesicles, when they fuse and merge with this presynaptic membrane, and as a result the release of neurotransmitters takes place into this synaptic cleft, and these neurotransmitters, they bind to and they activate the receptors of postsynaptic neuron, and soon they get diffuses away, and this transmitter is metabolized or it is transported back into the terminal knob. And when this postsynaptic membrane is activated, again the depolarization starts and this transmits the nerve impulse to the second neuron from this first neuron. So in the process of conduction of information as chemical synaptic transmission, first step is the action potential. It reaches or it arrives at the terminal knobs and it results in the depolarization of the presynaptic membrane. As a result, the voltage gated sodium channels, they open, and further it results in depolarization of the membrane. As a result, the voltage gated calcium channels open and the calcium ions, they flow into the presynaptic neuron from outside, because their concentration is greater outside the cell than inside, and when they flow inside, the synaptic vesicles, they migrate towards the presynaptic membranes, they merges with it and they forms a continuous membrane with presynaptic membrane, and they release the neurotransmitters into the synaptic cleft by exocytosis. This neurotransmitter, they diffuse across the synaptic cleft and they bind with the specific receptor proteins or the chemoreceptors present on the postsynaptic membrane, and this causes the sodium channels to open, and again depolarization starts. Like this, the transmission of a nerve impulse takes place from one neuron, that is the presynaptic neuron, to the second one, that is the postsynaptic neuron, through a chemical synapse. So with this I end my presentation. Thank you.